Hello, everyone. My name is Gabrielle Bizzaneri, and I am a senior at Westfield State University. I completed a senior honors project this year, and the title of that is Ingesting Secondary Microplastics Induces Apoptosis Without Proliferation in the Daphnia magna intestinal epithelium. My project centers around the plastic crisis and the effects that it's having on organisms in our aquatic environments. In particular, 5.25 trillion plastic particles are estimated to be circulating in our oceans at this very moment. 242 million metric tons of plastics and solid waste entered the environment in 2016, and plastics are projected to accumulate to 710 million metric tons globally by 2040. In particular, microplastics are a contaminant of emerging concern. Microplastics are characterized as being 5 millimeters or smaller in size, and they are also characterized by shape, polymer type, and their color as well. For our purposes, we wanted to look at secondary microplastics and the physical damage that might be incurred on the intestines of Daphnia magna, or commonly referred to as a water flea. These are small invertebrates found in freshwater systems. They're easy to grow in cultures and maintain in cultures. And so we wanted to use these as a model organism, as previous studies have done, to examine the effects that ingesting secondary microplastics has on them. And just to define secondary microplastics, those are plastics that have been broken down from macro or large plastics over time. And these plastics in particular are rugged and irregularly shaped and can also be very sharp, as opposed to primary microplastics, which are manufactured as spheres and pellets, which are usually rounded and smooth. So to move forward with this study, we developed methods to prepare microplastics manually, and we used a red fleece sweater as our source for these microplastic fibers, and used razor blades to cut them up in batches, and we used 90 milligrams for each exposure trial for a total of 2 exposure trials. In the first exposure trial, we raised 12 adult individuals, fasted them for 22 hours, and then exposed them, or half of them were in the control group, half were in the exposed group, and they were exposed for a total of 48 hours. In the second trial, we did the same thing to establish positive and negative controls regarding cell proliferation and apoptosis, or cell program death. So the 2 factors we were looking at in terms of cellular responses were proliferating cells and cells that were dying as a result of ingesting microplastics. And the way to do that was using a tunnel assay and BRDU labeling. And the tunnel assay was linking red fluorescent probes to degrading DNA that is characteristic of apoptosis. And in doing so, putting it under the fluorescent microscope made it fluoresce red. With BRDU labeling, this label was being integrated into newly synthesized DNA and then fluoresced green under the fluorescent microscope. Further analysis of our data included measuring the average length of the animals in both groups and the amount of cells undergoing apoptosis and cell proliferation between the exposure and control groups. For a quick overview of our results, the average fiber size of microplastics was 392.02 micrometers in the media column and 75.15 micrometers ingested. 80% of microplastics in the intestines of the exposed group fell within 0 to 120 micrometers. 25% of the column microplastic sample were 300 to 360 micrometers long. And this is displayed in Figure 2. Figure 2, Part A, shows the average length being very similar between both groups. And then in part B, it's showing the distribution of microplastic fiber length, which there is a small overlap between the two groups because we expected most of the ingested pieces to be on the smaller side as congruent with their anatomy. And then whatever was in the column of the sample that we took would be a little bit longer because they're not physically able to ingest that. And that just further confirmed our ideas about Daphnia being readily able to ingest microplastic in sizes that are congruent to their algal food source that they typically consume. Figure 1 shows whole animals in part A and C in the control and microplastic exposed groups, and B and D shows just the intestines dissected out of the body, and A and B is the control group, C and D is the microplastic exposed group, and in D in particular, you can see the plastic is very visible and the red pieces are all throughout each intestine that was dissected. For cell proliferation, we found that it was absent in the control group, and you can see that in figure 4, it was 0 cells undergoing cell proliferation in the control group. 1.3 cells on average were undergoing cell proliferation in the exposure group, but it was not a significant difference. On the other hand, apoptosis was observed in one of the 6 control animals and in all of the microplastic exposed group. And this is indicated by an arrow in figure 3, part C, where the arrow is pointing to a positive result in the control group. And part G of figure 3 is showing an arrow to a similar positive result undergoing apoptosis in the exposed group. And on average, the number of cells undergoing apoptosis was 7.5 positive cells in the microplastic exposed group and 0.1667 cells for the control group. This was a significant difference. So apoptosis was significantly higher in our microplastic exposed group as opposed to our control group. Plastic may have been retained in the intestine of Daphnia up to 48 hours, but further studies examining exposure and recovery periods are required to confirm this. The observed increase in apoptosis in microplastic exposed animals may be a result of proximity to injury, cellular stress response, or inflammation. Intestinal apoptosis induced by microplastic ingestion may have an energetic cost or impair nutrient absorption, leading to the increase in mortality and decrease in growth and fecundity found in previous studies. Our findings suggest apoptosis induced by microplastic ingestion may have broader implications for invertebrates and ecosystems on a larger scale. And with that being said, there is more research that can be done to further what we found in this study. This in particular looking at microplastics that are different sizes and shapes and different colors and seeing how Daphnia interact with that scenario. It could also be expanded to include food web interactions with predators to Daphnia, and there's just so much more that can be done to see how microplastics are impacting freshwater organisms. I just want to say thank you to Dr. Christensen, Dr. Parshall, and Dr. Giuliano for their support as my Senior Honors Project Committee. And thank you to the Honors Program for the opportunity to pursue this project. And with that being said, thank you for listening, and I hope you enjoyed.